Excellent. All yours. Okay, so hello everyone. I'm Matthew. I'm a master's student, and my project here is going to be about dynamic PET images and features that can be extracted from them through radiomics. All right, so introduction and background. Glioblastoma, I'm sure we're very familiar with this. It is a very aggressive, highly lethal tumor, 10-year survival rate of less than 10% and 15 months average survival time. We image GBM through PET imaging. And specifically, we have a number of options with regards to the radionuclide that we use to access those images. So in this particular case, we have options of FET, we have options of methionine, we have options of FEG. So importantly, these radiotracers function by exploiting the fact that tumors have preferential uptake of amino acids and glucose. And these tracers then mimic the behavior of those molecules and are taken up preferentially. I also want to delineate two different types of PET imaging, static and dynamic here. So static PET imaging is where after a uptake period, we then collect radioactivity in the patient over a specific imaging window. And from that, we yield a single 3D image data set, which is static and shows the spatial distribution of the radio tracer. In contrast, we have dynamic PET on the other hand, which is a sequence of 3D image data sets across the entire duration of the uptake period. And in this particular case, we essentially have one image data set per discrete time interval of the scan. And doing so, we can access the tracer kinetics, which will be of importance in a few slides. So from these imaging modalities, we can then extract quantitative features, which allow us to go deeper beyond visual interpretation to extract quantitative insights. And specifically, we can use radiomic features to build machine learning models for all sorts of things, classification, prediction, prognosis, and such. More on this in a moment. So specifically, static radiomics yields static features and dynamic radiomics yields dynamic features. And in static radiomics, we have a bevy of standard features, I suppose, such as first-order features, shape-based and texture-based features, which describe different aspects of tumor morphology, the relative spatial arrangement of voxels and their intensities and such. In dynamic radiomics, however, we have a bit more of a nuanced situation. We can have timable static features, which are essentially static features resolved across different points in the uptake scan across those many data sets I discussed previously. Or we can have higher order model-derived features, which are of particular importance. So in the dynamic radiomics workflow, we always begin from the image acquisition. We have a series of 3D scans. We pre-process them, and then we delineate regions of interest. From those regions of interest, we have a spread of voxels. And each of those voxels will then show a specific pattern of radio tracer uptake over the full duration of the scan. In doing so, we fit each of those voxels with a time activity curve, which essentially shows the uptake of that radio tracer weighted by the dose over that time interval. From that, then, we can do either full kinetic modeling or something a little bit more lightweight called a semi-quantitative parametric map. And these give us a way to essentially fit compartmental models to the THCs, which allow us to extract kinetic parameters, in this case K1, K2, which essentially describe biological processes which go beyond static features. So in this particular case, the influx and efflux rate of the tracer, the washout, metabolic trapping, and a whole host of other features. The idea is from these kinetic parameters, we can then extract a parametric map. Every voxel has a value of one specific parameter, and the map is simply the net total of those features, of those parameters. So in the feature extraction phase, we have multiple methods to approach this. We can have radiomics from parametric maps, which is the standard method I was talking about before. In this particular case, I must reiterate that there are two different layers of features here. The voxels yield kinetic parameters, which are themselves features. And on top of that, too, we can then consider the entire parametric map as in and of itself a 3D image. And in doing so, we can then extract higher order features, which are analogs of the static features I was talking about before. So first order, shape-based, texture-based. And in doing so, we can yield insights from the geometry of the map itself, rather than just, say, an individual scan. So these are of particular importance. We can also re-extract the standard set radiomic features that I discussed before through some frames. specific dynamic frames. We simply choose a particular point in the uptake where, say, there's maybe maximum uptake, perhaps, and we simply extract static radiomic features from that. So using dynamic radiomics doesn't preclude the usage of static features. It just gives us a more nuanced way of extracting them. And of course, there's pre-processing one can do to either of these stages, but that's more technical detail. All right, this is the really interesting part. So true 4D radiomics. So until now, we have been discussing a regime where every voxel has a unique kinetic parameter from a single TAC. When you arrive upon that kinetic parameter, you are essentially compressing the entire time interval of that whole scan into a single value. And in doing so, you can miss some details. Like, for example, if you have two different voxels with the same parameter, it could be the case that same parameter came about, say, from two different patterns of uptake or washout. And standard radiomics wouldn't let you determine that. But 4D radiomics gives you essentially a way to view the entire progression of the data set in 4 dimensions. So you get to see the specifics of the uptake and the washout, which might not be captured otherwise in standard scans. We also have deep radiomics. Deep radiomics is another area of active inquiry. Until now, we have been prescribing the features that we are looking for. We are telling the system specifically what kinetic parameters are of interest. However, deep radiomics allows the system to determine this for itself. We essentially allow it to learn the features that it should be extracting. This is something I do not know too much about myself, but I'll definitely try to explore given the time. All right, stage 7, feature selection. This is a very important step. Essentially, not all features are going to be relevant, reproducible, repeatable, some combination thereof. We need to cut down our total set of features to only those which are most clinically relevant. And then, and only then, can we train machine learning models from those resulting features. Once we have trained those models, we then need ways to evaluate, say, the efficacy of those features and models. And in such, we have, say, various metrics such as accuracy, sensitivity, the confusion matrix. Those of you who are familiar with machine learning will know these terms. Essentially, we can look for things such as the area under the curve as a metric for discrimination, say, between molecular subtypes. And in the case that we're trying to prognose, say, some outcome, we have tools such as the Kaplan-Meier curve and the log rank test. These are well-established in literature. I'm sure those of you who have written papers in radiomics have used these yourselves. All right, so why is it important? So, as I was alluding to before, dynamic features seem to be particularly effective in predicting certain mutation statuses, in this case, a terpene mutation status. However, when used in conjunction with static features from dynamic subframes, we actually see additional insight for, say, other types of molecular code deletion. In this particular case, literature unveiled that 1p19q code deletion required a combination of static and dynamic features for maximum insight. Secondly, patient management. This is extremely relevant to me, actually, in my research. Essentially, when combining static dynamic PET, we actually notice a very strong increased efficacy of prediction. Particularly, Samini's paper was rather important to me, as it showed that certain methodologies have to be incorporated when accomplishing this, which I will go into some more detail about when I talk about my research plan. And third, we have accelerated clinical strategies. It is of no wonder that dynamic PET scanning is non-invasive. And because it's non-invasive, we are not wasting money, man-hours, and resources, and potentially patient risk and discomfort on things we don't need to do. So essentially, the idea is the consequence of this will be the development of a standardized pipeline through which this can be implemented at a much larger scale. And in doing so, we can optimize treatment plans, particularly for dose escalation, identification of particular zones within the tumor of interest, or simply just improve target delineation. All right, aims, my specific aims. Number 1, we will develop and validate a computational pipeline for extracting features at the voxel level. Secondly, we will identify a subset of these features which are resilient, stable, and reproducible for clinical application. In doing so, we will then extract the pipeline to a second higher quality data set, a much larger one too, sourced from a national multi-center clinical trial. And then, and only then, in the longer term, I will then try to use these extracted features as inputs to machine learning models to improve the prediction of outcomes specifically. So this is my research timeline. Initially, of course, we are given 2 data sets. The smaller one, we also have test, retest, and multi-observer components. And of course, we have our larger data set incoming. Phase one is always the pipeline development. We essentially extract the parametric maps. In doing so, we have to handle pre-processing. There is a whole chain of thought about this, which is its own subject. And in doing so, we can implement models, kinetic models appropriate to that parametric map. And from that then, and only then, can we extract the features. We'll use radiomics to do this. You can see here, this is a brief summary of the procedure I will use. The possibly most complicated part might be the segmentation ROI mask, but I trust that my various colleagues will yield some advice to me as to how to treat that, I hope. And as a result, we get an ordered dictionary of features. That's very important. So next, we're going to check the reproducibility and the feature stability. Here, we are looking for stability under both defined conditions, which are conditions we can control, such as are there different observers, or is the setup identical and repeatable, and also intrinsic variation, which is things we can't control. In this case, scattering noise, intrinsic positioning differences, and such. We have various such metrics to determine this. Essentially, what we are doing is, for every test, retest, and inter-observer data set, we are looking to calculate each of these metrics for each pair of features in those sets. That will give us an indication of the stability of those paired features. And we can visualize them with Flat-Outman plots, for example. And ideally, we receive a table of all of these metrics for each feature. And it will give us a way to essentially quantify how effective are these features in a reproducible sense. And essentially, we will assess these features by simply demarcating a threshold. Let's say for this specific metric there, the CCC, we'll say all features above, say, 0.85 on that score will be selected for the final outcome. So of importance as well, multi-center reproducibility. We need to keep in mind that the multi-center trial may incorporate different scanners. And in doing so, of course, you need to take into account the fact that there are going to be differences inherent to that. So combat normalization, demonstrated by Samimi, a familiar name, offers a way for us to essentially attenuate this effect. Phase 4 is feature selection. To be quite brief, this is where we essentially perform dimensionality reduction on the selected features to ensure that only the most uncorrelated and most predictive features remain. So the idea is we can use redundancy reduction using Pearson correlation. That's a way to essentially measure linearity in a direct sense. We can also group stable features using unsupervised clustering in this particular case. And once we have done so, then we can use statistical filtering, which is supervised feature selection, to only select those relevant to a clinical endpoint of consideration, which in my particular case is outcome prediction. So there are univariate tests, which are the t-test and ANOVA, for example. These just compare feature means between groups. And of course, we have non-parametric alternatives in the case that, say, our data doesn't fit the requirements to use the t-test and ANOVAs. And then, of course, we will use recursive feature elimination, which is more of a machine learning way to cut down the total number of features that we receive. And then finally, we can use techniques such as principal component analysis to essentially transform that output into an ordered set, and then only take, say, the first few features in that set, proving to be the most predictive. So a very big area. Phase 5, evaluate the added value of dynamic features. I trust that they will be of greater utility than static features alone, and in doing so, we can build new outcome prediction models. There are various frameworks with which we can do this. In this particular case, scikit-learn, XGBoost, familiar names, well documented in the literature. And of course, we can evaluate them using the methods I described before. Of particular note, computational complexity and processing time. Of course, the data sets at play here are very large, and especially for the multi-center data sets, there will be a lot of time required to generate these maps and extract the features. And it is my hope that in the process of this research, I can also determine ways to optimize the existing pipelines, perhaps even in radiomics itself. Especially when using four-dimensional radiomics, the native libraries aren't so particularly supportive of that level of analysis. So I will have to do some custom scripting myself. It is my hope this entire process will take about 13 to 15 weeks. I prefer to overestimate than underestimate. It gives myself some wiggle room. And essentially, I have no doubt I'll call upon the expertise of not only my dear audience, but also my friends in the United States who may or may not be watching me right now on Zoom. So last point, alpha evolve. Who here knows about this? Who's heard of this? came out a couple of days ago. Google released this. You familiar with AlphaFold? Protein folding? Yes, of course. Same team, Google DeepMind. So they just released a new generative AI tool to iteratively improve algorithms. This is of particular interest to me. Specifically, when we fit computational models to the TACs, there's a period of nonlinear curve fitting, which is computationally intensive. It takes up a lot of the load, if you would. I'm thinking I could use AlphaEvolve to potentially streamline this process. I actually signed up for one of the academic rollouts of this tool about 3 days ago or so, and it's my hope that once I do gain access, I can run some tests in my own spare time to see how perhaps it could expedite the process. So that is an area that was not in my literature review or proposal because it just came out. So there we are. And that's my proposal. So thank you for being a wonderful audience. Thanks for listening. Any questions? Yeah.
Thanks, Matthew.
Yes.
Radiomic stuff. I was wondering if we could maybe step away from the radiomics stuff and sort of ask a simpler question as to why are we looking to use FET-PET for glioblastoma patients? Because it's not standard clinical practice. It's a trial going on to see if it's good or not. Because do you know what imaging that they usually use for glioblastoma patients?
Well, in literature I've seen, usually it's FDG. Actually, FDG and mostly those two.
Yeah, I think it's more common for brain cancers or at least every brain cancer will have an MRI image. Glioblastomas are assessed during follow-up using MRI only. FDG can be used. I think often it's as year-round diagnosis, but obviously there's some issues with FDG based on the background uptake and how this will tumors. So I think that's also something to just consider as to maybe why we're sort of looking to use this new imaging technique. And you were talking about how, that tumor segmentation and, you're asking they might need a little bit of help, but do you know how, because where you extract your features, you're going to need to define area to extract them. So do you know how the methodology we use to extract to segment that region?
Actually, that'd be really great for you to tell me. I'm familiar with the computational process, but also like the fundamental manual way to get it done.
Yeah. So what we usually do is we'll put like a bit of a region of interest where we believe like suspected tumor areas, and then we'll take a background assessment. So on the other side of where the tumor is, we'll take an assessment of healthy uptake and then compare it to the uptake we see in that's around that tumor area, and then there's a threshold which is a 1.6 tumor background ratio, which will then you can threshold that bigger volume that just sort of encapsulates the area which should be tumor infiltration. You won't have to worry about segmenting that yourself because we leave that to nuclear medicine physicians. And so, but it is good to be aware of how that process is done because that's basically following sort of the guidelines that are set out by the community so far. And so, would you go to the slide in terms of the data availability?
Yes. Which one was that? Here?
I think it was just the next one. Yeah.
This one?
Yeah.
Okay.
So I know the big trial, there's a lot of data coming through. I would just sort of take into account potentially not getting all of that data by the time you sort of are starting to do your research because it's data collection is a bit of a notorious process and it gets a little while. There is that initial data set that is here ready to go, which is great. In terms of test-retest imaging, there's about 9 patients that have that test-retest imaging, and for the multi-observer, there's about 6 of them. So out of that total number of patients, so that's also something to be aware of about the, I guess, statistical power you might have. It's still definitely something worth exploring, but that's going to be a limitation that you're just going to have to address in your work.
Absolutely. That's actually an implementation point for 4D radiomics. 4D radiomics has shown in literature I've seen to be particularly effective when the available data sets are not as expansive. So, yeah.
All right, that's all from me. Thank you.
Thank you.
Cool. Does anyone else have any questions from the room?
If not, we have someone from Teams.
Obviously.
Yes, being a practitioner, also a researcher, how do you envision the integration of the dynamic PET radiomics into the current clinical overview for the GBM management, particularly regarding the decision-making, treatment, adaptation, and stratification?
Okay, so in particular, great question, by the way. In particular, there is a well-documented advantage of the fact that dynamic PET is non-invasive, and moreover, it can be automated at particularly large throughputs for a large number of patients, especially when you have a developed healthcare system. So what I hope is once I develop an established pipeline that reaches all benchmarks, that this can essentially replace, or at the very least supplant, the rather I'd say discomforting process of surgical resection. Because typically you have to do biopsies to say assess molecular type or maybe extent of the spread. Hopefully with this we can just completely bypass that step. But it is of course contingent on the nature of the insights that we receive. Because as you know, multiple types of glioblastoma are the same. There are many subtypes. Some might be heavily predicted, for example, by my features, others might not. So it has to be done on a case-by-case basis, but I'm very happy that this seems to be appealing enough for you to ask such a question. If I can also ask, can you tell us a bit about yourself?
Yeah, I'm an oncologist. I'm also a researcher in the US, New York City.
Wonderful.
Thanks very much, Dr.
Thank you.
Brilliant. Thanks very much. Any other questions?
I'll jump in very quickly because I know we're strapped for time. So you've mentioned a lot of features, and I guess there's substantial potential for this to explode into thousands of features potentially. And of course, your end goal is outcome prediction. So I guess twofold question: what outcome are you actually trying to predict? Although I guess potentially that's a PhD question for you. And secondly, given the size of the data set that you have, do you have a approximate goal of the number of features you might want to include in your final model to avoid overfitting?
Right, that's a great question. So to begin with, there's a very large step about right here, where just a priori we're going to be excluding a very large number of features based on whether or not they meet a specific criteria on the metric. And then post that, we will then take the remainder of those features and run them through various forms of, say, PCA to just reduce the dimensionality even more. So truth be told, I'm very curious how many features we'll end up with as a percentage, because this will be very, very informative. If, say, in the N=27 data set, we were to run this whole process and receive maybe a handful of features, that might not be sufficient. But in a much larger data set, that might not be the case. So we'll see. To answer your first question as well, so the outcome that I will be looking for, the continuous outcome might be survival time. So just say expected survival time just as a continuous variable. In a continuous variable, that might require different tests, actually. Some of these are bivariate. Of course, there's always the outcome prediction of do they survive or not, and that's a lot easier to work with in a statistical sense. So yeah, I think ideally that survival time in the continuous sense.
Cool. So you're excluding features that were non-repeatable from the FET study.
Correct. Yeah.
That's the same data set though as this one. Is that
correct? Yeah, and that's why hopefully I can move to the larger one.
Okay. So the plan then is actually not to do any repeatability analysis. It's just a reproducibility.
Yeah. Okay. So the thing about repeatability and reproducibility is that, to tell you the truth, they're actually very highly correlated in a sense. Because if you can show that, say, a feature is stable to the identicality of a setup in test-retest, you can also show that there was a very significant association with inter-observer stability. So in a way,
I get that, but there could be features that are non-repeatable but are reproducible because you're just testing different things. Right? Right. So your reproducibility analysis will be with respect to different observers. But cool, thanks, Paul.
Yeah, thank you.
Time to move on then. Brilliant, thank you.
Well done.
Yes, there was, Emily has got a question. Emily, if you question.
Yeah, I
Yes.
just had a real quick one. I'm just wondering if there's any thought to like any commercial interest with these models, and if so, what's the implication of using programs such as Alpha Evolve?
Mhm. Yeah, so sorry, repeat your question.
No, that's all good.
Yeah, okay. Yeah, well, if you're talking about, say, the commercial applications of what I'm doing, well, fundamentally, it saves a lot of money. And I think that's the bottom line for business applications, or even just in healthcare. I think the fact alone that this can be fully automated with essentially no oversight, given that we specify the correct features, I